PhD thesis. He came to us from the University of Coimbra in Portugal. I learned that it's the 6th oldest university in the world. Bologna is the first. I'm not going to say any more about it, but I'm going to show you about it. So let's have the first. You see him in all his disguises. The one in the center, on the bottom, is disguised as a cowboy. I think the one at the top on the right is a Patriots football player. Next slide. Here he is with his 2 advisors. I don't know who this one is. This one is Chad Cowan on a fishing expedition. Next one, please. He went to many meetings. You have to shut him up and you can see someone has a particular way of doing it. Next slide, please. This is a very interesting slide. You have good taste. This one is Brianna Johnson, who was an undergraduate student. I hope you'll forgive me, but I thought I should send a copy of that to the dean. So that's what I did. This is his current student, Andrew, who's here. Next slide, please. Oh, yeah. He traveled widely during his graduate career. He participated in a science course in Bolivia, organized by the Bolivian Science Club. There he worked on a cure for AIDS. I don't think he knows what AIDS is. Next slide, please. I had told him that before he got his PhD, he was going to have to learn how to horseback ride. So here he is learning. I think that's in Bolivia. Is that right, Paul?
Yeah.
In Bolivia, Mo's father runs a ranch in Bolivia, which they visited a few years ago. Here he is winning a ping pong tournament, dancing, and on a water slide. Next one, please. Oh, this is how we'll all remember it. And a full one waiting in the background. Next one, please. There he is, Leo and Mark. That's the signature for his emails. Well, the next one's gone.
I couldn't play the video, but I can try.
Where's the dancing one?
Good afternoon, everyone. Thanks for coming. Thank you, Jack, for the kind introduction. I'll just say that I brought my own collection of photos myself at the end. So every single person in this room is here today thanks to successful pregnancy. And we take pregnancy as something very natural that obviously works. We couldn't think of it being any other way. But from an immunology standpoint, pregnancy is actually a paradox. Because if you think about it, during pregnancy, there is a fetus that is 50% foreign to the mother. However, the maternal immune system does not reject it for 9 months. And the first scientist to put this paradox forward formally was Peter Medawar in the 1950s, at a time where the basic laws of transplantation were being laid out. And this would seem to violate those basic laws. So several options were on the table. At the time it was thought, well, maybe pregnant women are systemically immunosuppressed, so they will not reject the fetus. That turned out not to be the case, because pregnant women can fight viral infections. In addition, if you transplant a foreign organ into a pregnant woman, she will reject it. So it's going to be something local. So there's another option. So being local, maybe the fetal cells are somehow shielded from the maternal immune system. They don't come into contact. But as it turns out, that's also not the case. Fetal-derived extravillous trophoblasts deeply penetrate the maternal tissue and remodel arteries at the implantation site. And this implantation site is very far from being devoid from immune cells. Actually, there's many of them. And these cells are natural killer cells. So unlike in the blood, where most of your immune cells are T cells, at the maternal-fetal interface, 70 to 80% of your immune cells are natural killer cells. And so you would expect these natural killer cells, which are usually involved in killing virus-infected cells and cancer cells, to recognize these foreign cells and attack them. And that does not happen. And we still don't know very well how this local immune tolerance induction occurs. But we do know of one gene that is specifically expressed by these extravillous trophoblasts. And that gene is HLA-G. HLA-G was first discovered in the Strominger lab many years ago in a southern blot in the times where the MHC locus was being discovered, the major histocompatibility locus. But unlike the most MHC genes that you've heard of, HLA-G is not involved in communicating with T cells to trigger killing. It's also not polymorphic. And unlike other MHC genes, it's very restricted in expression. It's almost only found in these extravillous trophoblasts. And finally, it's not an activating ligand, it's actually an inhibitory ligand. It's able to inhibit NK cells. So we have this gene that is specifically expressed in this set of fetal cells that invade the maternal tissue and deeply mingle with maternal immune cells without being rejected. So the question is, how is this gene expressed in these cells and not in other cells? So we know that all MHC class I genes have very similar classical promoters. And so they have a very defined set of motifs that usually work. For HLA-G, however, even though those motifs aren't there, they're actually non-functional. So we don't know, so this is not the answer to understand the expression of HLA-G in trophoblasts. If anything, it tells us, well, most cells do not express HLA-G because this promoter does not work. So we have to look somewhere else. So we know that genes are usually regulated by non-coding regions, which are called enhancers. And so we set out to discover these new regulatory regions. And so traditionally what we do is have a candidate region, and we do a reported gene assay, where we clone that region upstream of a reported gene, say luciferase, and then you measure it until we find one you want. But it'll take a really long time. If you look at the locus, it's roughly 30 kb. That'll take many, many years of grad school. So Chad actually told me, you should call this really smart guy, Tarjei Mikkelsen, at the Broad Institute. And so what Tarjei did is that he came with a reported gene assay version that is times 12,000. You can test 12,000 oligos at once. And so the way it works is that you'll synthesize 121 base pair long sequence of DNA that it can cover the entire locus, styling it, and then you'll clone them into a complex library where the plasmids have the cloning sites, have a synthetic invariant promoter, a luciferase, and importantly, an RNA-seq tag. What this means is that each one of these 12,000 different plasmids has a different sequence. So if you were to transfect this clone library into your cells of interest, and then you sequence the RNA from them, you can then trace back the increase in input in RNA to the particular element of interest. And so what this technique gives you, it's an element resolution. So it gives the resolution at the tiles across the locus. So that's what we did. We tiled the entire HLA-G locus, which we defined as this 27 kb region. And so we asked what happened. And so you can see here that we built the library, and then use JAK3 cells. JAK3 is a cell line, there's a trophoblast cell line that expresses HLA-G, and is using the field as an in vitro model for the extravillous trophoblast I just told you about. And so you can then transfect these cells and then sequence the RNA to see which elements increase the output of that invariant promoter the most. And that would be an enhancer. And so with all these 12,000, surprisingly, it did work. And so on the top, you see the results. There are squares and circles, because for greater confidence, we actually use not one, but two promoters. We use the weak minimal promoter and use a strong SV40 derived promoter. And so the results on the y-axis is enhancer activity, which is defined as the increase in RNA output compared to the median of all 12,000 tiles. And so you can see in that pink rectangle that there's some action going on. And in particular, the most confident candidate, this candidate number 1, you see it agrees very nicely between both promoters and is very high in both. And then here in the bottom is a simplified view of the top chart with just the top 3 candidates. And so next, I clone them individually in the classical luciferase report gene assay I told you about. And then from all these 3 or 4, one of them, number 1 in red, was one that really got validated as a true enhancer that can increase the promoter activity in at least in this context. And then the next question was, well, we're looking at cell type specific expression. Is this enhancer cell type specific? So in this case, use a different cell line, 293T cell, which is a kidney cell line that does not express HLA-G. So in theory, it shouldn't work, and it didn't. In addition, we actually took an extra step, and we actually inverted the sequence of the enhancer and asked if it still works. Because by Dashner's traditional criteria for an enhancer, it does not depend on the distance. So you see this enhancer is all the way 12 kb upstream of HLA-G, number 1. Number 2, it should not depend on the orientation. And so you see that even if you invert this enhancer sequence, it still works, specifically in the HLA-G positive JAK3 cells. So we're confident we found an enhancer. At the same time as the experiments were going, we also decided to look at this region in this endogenous chromatin context. And so to look at that, we did DNase sequencing. And what this means is that you use DNase to digest chromatin, and this way you can find which regions of the chromatin are more open. And openness correlates to transcription activity. So if you have an active enhancer, you'd expect to see a spike in the chromatin openness. And that's what we saw. If you look at the whole HLA-G locus like we saw before, you see that there is a spike precisely around that region where this enhancer sits. And for this, we call this enhancer L for long range. In addition, we also see some CTCF binding sites. And CTCF is a very interesting molecule because it works as an insulator. What that means is that it's been shown that it's involved in chromatin looping, and it isolates the activity of enhancers to their particular neighborhood. So this suggested us that the enhancer will act on this gene and nowhere else. We'll see if that's true. So far I have very nice evidence that this might be an enhancer for HLA-G expression. But I haven't shown you any single piece of data on HLA-G expression. So that's what we did next. So the question here is, how can we assess, how can you assess the role of this 121 base pair piece of DNA 12 kb upstream of a coding region, see if it has an impact or not? And these questions be pretty hard to address. You'd have to clone many variants with different point mutations and see where it breaks down and try different cell lines. But with the advent of genome editing, this actually became feasible. And so we did this using CRISPR-Cas9. And so very quickly, CRISPR-Cas9 is a system with 2 components. We have a Cas9 and nuclease that has a machete and does double-strand breaks where you want. And you decide where it goes by having a guide RNA that is base complementarity to the sequence of interest you have. So you can basically guide it anywhere you want in the genome. And so usually people then, make different mutations, endogenous mutations, and see what happens. Well, we decided to be more of surgeons. And so actually had 2 guide RNAs, 1 on each side, and we asked what happened if you'd excise this enhancer from the genome. So it's clearly developed in New England. So you go with your Cas9 with your 2 guides, and then you excise your 100 base pair region, and you can really ask what happens. And so first things first, this system did work. And because this is a cell line, it can actually have individual clones derived from single cells. So I can show you independent wild-type clones and knockout clones that have the deletion that I expected. So these cells are homozygous for this deletion. They do not have the enhancer. So what does that do to HLA-G expression? It turns out that this deletion does not decrease HLA-G expression. It completely abolishes it. So you can see here in orange that all the knockout clones, their HLA-G expression, you analyze by FACS. So to look at surface protein expression, so the most stringent tests of HLA-G expression, you see it's completely gone. If you look at the wild types, which underwent the process but there was no cutting involved, you see they're just like parental. So this is evidence that this enhancer is absolutely essential for HLA-G expression. And then we confirmed this data outside the message level with quantitative PCR. But we wanted more. We wanted to know, well, maybe you delete this piece of DNA and there's something opening at the locus level. Because chromatin is a very complex structure. Maybe we're messing with other kind of aspects, like matrix attachment, etc. So we want to know if anything else changes, if this enhancer is specific. And so to address this question, we teamed up with John Wynn's lab, in particular with Bill Mallard. And so what we did was, in panel A, you can see at the RNA-seq level that the wild-type clones all have the nice reads, each exon. The knockout clones have no expression left, it's a complete knockout. You can see some blips there, but please note that the scale here is 0 to 700, while here is 0 to 10. So we confirmed that the knockout is perfect. In addition, we tried to look elsewhere. So if you look at the neighborhood of 2 megabases to the left and to the right, and it shows this distance because the furthest acting enhancer reported acts 1 megabase away from this gene. So 2 megabases we thought pretty safe. In addition, this neighborhood you'll notice includes all other HLA-G genes, all other HLA genes. So you can really see if all HLA genes are affected or not. And they were not. So HLA-G is by far the only significant difference when you delete this tiny piece of DNA. And then finally, since this experiment was done transcriptome-wide, we can look at the whole genome level, and if you compare enhancer 1 knockouts with either enhancer 1 wild-type clones or with the parental cells, you see how HLA-G is far to the left, so it's a big decrease, and nothing else really reaches that level. If you look at parental versus enhancer 1 wild-type, so parental versus cells that underwent the process, but there is no cutting, you see that nothing changed. HLA-G is right in the middle. So we're very convinced that this enhancer L specifically acts on HLA-G expression in trophoblast cells. Were we though? Because the problem is that this JAK3 cells is a cell line, and more than that, it's the choriocarcinoma cell line. It's a type of developmental cancer. And for one, it's a cell line, so it might not fully represent physiology. Number two, I didn't tell you, but actually some cancers have learned how to express HLA-G, in particular melanoma. And so they use this as a way to evade the immune system. So you could argue that maybe I found an enhancer important in cancer expression for HLA-G, but not on trophoblast expression for HLA-G. So we want to ask if the finds that we found, if they hold true in primary trophoblast cells. And so we're lucky that in the Strominger lab, we get pregnancy interruption samples every Tuesday. We have a whole team. Our lab has developed a method to purify HLA-G positive extracellular trophoblasts. And the team with Tamara and Anita and Vita and Angela is not pictured here. And so I was able to repeat this work in primary cells. So it was not easy. These cells only live for 2 or 3 days in culture. We don't fully understand how they grow best. And so, however, in addition, you can't just transfect them with DNA. I had to use lentiviral. So if I transduce them, 4 times, 12 hours apart, you get some cells that are GFE positive. And then if you look at these cells and look at the mRNA expression level of HLA-G, you do see that HLA-G expression is significantly decreased. In addition, if you look at the protein expression level, we also see a decrease as a response to deleting enhancer. And I should tell you that HLA-G is an unusual MHC molecule for several reasons. One of them is that it has a very short cytoplasmic tail. What that means is that its turnover is very slow. So this is, after 3 or 4 days, this is as much as HLA-G will turn over after that time. So this is a very significant difference. I was very happy to see this. So what I'm basically showing you is that enhancer L is essential for HLA-G expression in trophoblasts, especially in the cell line and in primary cells. So the next question is, how does it work? So we know, or the current state of the field will tell you that long distance enhancers will loop into the promoter of the gene they regulate, And this is due to different factors. There's squeezing, there's CTCF sometimes involved as well, and there's transcription factors that mediate that looping. So our first question was, the enhancer is very far away from the gene. Does it loop into its promoter? And so to look into this, we use this technique that is called 3C, chromatin conformation capture. So what it does is that it will freeze the chromatin set of a nucleus of a cell by using PFA, and then you can digest that DNA with the restriction enzymes wisely chosen, such as restriction sites flanking your regions of interest, and then you can do a PCR and find if there's an interaction or not. And that's very represented here in the schematic. These are two regions, enhancer L and the classical promoter of HLA-G, the restriction sites, and then the primers. And these primers are pointing outwards. What that means is that if you have undigested DNA, it will never work because the primers are not pointing inwards. But if there is a ligation product, this hybrid piece of DNA, then you'll get a PCR band. And so this is what I show here. So if you have undigested cells, you see no signal whatsoever. And to the left is the control ovary with a black primer, so that should always work if you have DNA. If you look at 293 T cells, which do not express HLA-G, you see nothing. However, if you look at JAK3 cells, which express HLA-G, the trophoblast cell line, you see the interaction is right there. And then if you sequence this, you see very neatly the DPN2 restriction sites, and then your enhancer, another restriction site, and glue to the promoter. So you basically find this hybrid piece of DNA as you'd expect. So this showed us that enhancer L, even this 12 kVA part of the HLA-G, it indeed loops into the classical promoter. And so this must be involved how it performs its function. So the next step is to see what are those transcription factors that are mediating enhancer L function. So my first attempt at it was to just chop up this enhancer and see if I can find a minimal region, or a small region at least, that is sufficient for its activity. I was, as you can tell, very unsuccessful. There's no surface activity in any of these regions. So what this tells you is that there must be multiple binding motifs that are spread all over its length. So what do we do at this point? We call Target again. And then this time we do the same kind of experiments, but this time your 12,000 pieces of DNA is just enhancer L over and over and over again, but we replace every single base pair. And you also add some small insertions and deletions. And so this way, we don't have an element resolution, we actually have a single base pair resolution. We're taking every single base pair and asking what happens. Again, with the same sequence, same readouts. And so this is what we found. This is a map where on the y-axis is mutual information. What that means is the higher this value, the more important the base pair is for activity. If you want a more visual display, you have this heat map here. You see that yellow is very hot and 0 is very cold. And so you see, for example, here in this high peak, that if you have an A, that's the only base that works. If you have any other base, you see how it's very dark. And so with this kind of approach, you're able to find 5 motifs roughly that seem to be important for enhanced activity. Otherwise, it just completely loses it. And then these motifs actually turn out to be binding sites, if you're doing silico analysis, for GATA and CVP factors. And these factors are very interesting for multiple reasons. But first, I should just tell you that this is not good enough. There is several family members of each one of these transcription factor families. And so what we did next was to try to narrow down on factors that are actually expressed by these cells. And so we used 2 lines of evidence. One was microarray data from primary EBT, side by side with JAK3 cells. So you can ask which factors are expressed. And then we combine that with our transcriptome wide RNA-seq data. So we know very well, we have a whole catalog of every single gene that is expressed in JAK3 cells. So if you compare those 2 pieces of data, you have 3 candidates emerging. You have CVP beta, you have GATA2, and you have GATA3. But there's more reasons to narrow down on these factors other than just these expression levels. If you look at total human gene expression across different tissues, you see that both CVP beta and GATA2 are enriched in placental tissue. It's this brown bar here with the arrow pointing at it. In addition, actually the third factor, GATA3, which is more known at least in immunology as being involved in CD4 T cell development, was the most tissue-specific of all observed. So you see it's by far more highly expressed in placenta than anywhere else. And actually there's a talk today from 12 to 1 that shows how this is an excellent marker for human true factor there. So in addition, if you look at mouse studies, these factors have a role in pregnancy, specifically in placentation. So both CVP beta knockouts and GATA2 knockouts have tremendous defects in placentation, in particular neovascularization. And that's why you see there's abnormal structure, and then there is less endothelial cells being recruited. So this mice have problems in placentation. And actually, CBP beta, no female mice are sterile. They cannot get pregnant. So also, finally, from a mechanistic point of view, the GATA factors are very attractive candidates because they've been shown before to be involved in chromatin looping. So this could be our perfect placenta-specific transcription factor that is involved in chromatin looping. In this case, of enhancer L. You even see a crystal structure, and you see GATA factors have 2 zinc fingers, and they use those to bring together regions of DNA. And so in essence, that's true. Do these factors bind to the enhancer? And so that's what we see here. If you do chromatin immunoprecipitation followed by qPCR, you see that these 3 factors, CBP beta, GATA2, and GATA3, are enriched at the enhancer. And then if you use other regions like the hemoglobin promoter, they're not there. In addition, what was interesting is that if you look at HLA-G promoter, which does not have GATA2, GATA3, or CBP beta binding sites, it's also enriched there. And what this suggests again is that there is a looping going on. So in the chromatin precipitation, you take out the whole loop of DNA, and then you still see some enrichment in that area. And then as a functional test, we expressed these factors, and that's what happened to HLA-G expression. So should they bind there, do they affect HLA-G expression? And they do. So if you look at X2, which is a typical trophoblast transcription factor, nothing happens to HLA-G expression. However, if you look at these 3 factors, all of them increase significantly HLA-G expression. And then one thing you could ask is, where does enhancer L come from? Like, how did it come about? And so it turns out that this enhancer L, if you look at its sequence, it has a retroviral signature. And so what that means is that viruses have been integrated in our genomes across evolution. And if you look at this enhancer, it's very well conserved in apes. And then if you try to look at rodents, it's not there anymore. And this is interesting because it's part of a lineage-specific retrotransposon. And these elements have been shown to create novel tissue-specific enhancers. This is because these viruses originally arose and they try to hijack a cell's machinery to being produced. So over time, if these viruses are, if they are inactivated and they happen to be close to a gene, and if they have the right transcription factor binding sites, and if that gene is somehow good during evolution to be expressed in that cell type, you can now gain a new cell type specific transcription factor binding element. And so, interestingly, if you look at it, all the great apes are there except for orangutan. So orangutan does not have enhancer L. If you look at its version of HLA-G, it is highly polymorphic, and it does not seem to be involved in immune tolerance. So enhancer L and the function itself of HLA-G seem to be together. And orangutan, and we know that orangutan diverged from our lineage 15 million years ago. So you can tell more or less when enhancer L arose. If you look at MHC genes, they're over 500 billion years old, if you look at it. Okay, so I hope I've convinced you that HLA-G is an MHC gene that, unlike all others, is involved in tolerance induction and is tissue-specific in its expression. And this is not achieved just by the classical promoter. There's actually a cell type-specific enhancer that, through chromatin looping and trophoblast transcription factors, achieves HLA-G expression. And so, once you have this knowledge that is still not complete, we can think of developing new contraceptives, or the other way around. If you tackle this system, you can improve on diminishing recurrent abortions. But if you think a little bit bigger, you can actually think of it more in a broader scheme from the immune tolerance perspective. And so right now, if you look at regenerative medicine, the current paradigm is you take a person biopsy, you make its own pluripotent stem cells, induce pluripotent stem cells, and differentiate the cell types you need, and then put them back in. And then if that works successfully, the person is cured. But this process is long, labor intensive, might not work. It might actually produce cells that are still triggering an immune response. So the idea or the dream would be, just like you have a universal blood donor type, if you could also have a universal donor stem cell. And so if we tweak pathways such as this HLA-G pathway and others, we might be able to engineer such stem cell. Okay, but going back to the puzzle of immune tolerance during pregnancy, I've shown that we're able to change the genome of trophoblasts. It'd be really nice if you could also change the genome of the immune cells that are involved. And so with this current explosion of genome editing, we thought it was a natural next step. But it turns out that back at the time, in the summer 2014, amazing progress in the field, but no one had shown that it could use CRISPR-Cas9 in primary immune cells. And so we just went after it ourselves. And so the first thing we did was we picked this gene, 32 microglobulin, which is a small protein, but very, very important. Without this protein, you cannot have MHC class 1 surface expression. In addition, from a pragmatic standpoint, it's a small gene, very small first exon, and all cells express it. So this was our ideal platform to test our CRISPR system in immune cells. But first, we'll just look at the cell line. So you see we saturated this first exon with different CRISPRs, and that's different activities measured by loss of beta 2M surface expression. So this is at the protein level, this is at DNA level. So it's called survey assay. If you see bands appearing, it means there is cutting at the DNA level. And so that's just a bar chart of that. So we're happy. You see there are several CRISPR sequences that seem to act very effectively, and it's at least in the cell line. So, at a time where so many people talk about building a wall, we're actually very happy about taking down the wall, the immune barrier wall. And so this would be one such tool as part of that future toolkit. To our dismay, however, once we started playing with human immune cells, in this case from primary blood, from peripheral blood still, we didn't see anything whatsoever. So something that worked really, really well in cell lines did not work at all in immune cells. No wonder it was not out there yet. So then we actually looked across different guide sequences, and you always see the white bars are efficiency in 23 T cells, so sky high. Equally efficiency in T cells, very, very low. Nothing is happening. So what we thought was, well, we have this dual CRISPR strategy. Can we apply it to also knock out genes? So you can flank an exon and excise it. Does that somehow increase the efficiency? It could increase. So what you see is that if you try different pairs, again, most of them don't work. We finally got one of them that does work. And you see the same kind of loss of beta 2M surface expression. We also confirmed the consequent change in MHC class 1 expression, at least with this pair. So it seems to be possible to engineer immune cells, but it's still not very efficient. So this does not seem to be the step forward for us for a first proof of principle. So we teamed up again, in this case with Eric Cross's lab, and in particular worked with Pankaj Mandala, postdoc in his lab. So the idea was that how do we make this, how can we come up with a better functional test? And so the cell type that we want to engineer from the blood is the hematopoietic stem cell, the HSC. And that's because the HSC, it can divide indefinitely, it can generate all blood cell types. So my T cells, which love dying, I can tell you that. And so in addition, this has been very established in the clinic. Bone marrow transplants have been doing for decades. So we tackled it, and then we went to the single gene disease. And it turns out that HIV or AIDS, they're not to be such a case, because there is a very rare population in Northern Europe that has mutations in this gene called CCR5. CCR5 goes for a receptor that is essential for HIV infection of CD4 T cells. And some people are lucky enough, they won the genetic lottery, per se, that they have mutations in both alleles and they are resistant to HIV. And there's actually been a patient, Timothy Ray Brown, which was cured of HIV this way. So can we use genome editing to basically make everyone win a genetic lottery? And can we use that as a model for our CRISPR editing? So we teamed up with Pankaj Mandal from Derek's lab. And then you can see here is that if you use the same dual guide approach, but this time in HSCs, you see an amazing deletion efficiency. So if you look at this gel here, there's a wild-type band, primer's flanking it, and then the bottom lane is a knockout band. So if you lose a piece of DNA, it will go lower. And you see that these are single-cell derived clones, because HSCs, again, they can double forever. And you can see very nicely that there is several clones that are just straight knockouts. And actually, if you play enough with the pairs that you use, we found one that gives you up to 50% of gene editing efficiency. So that was something clinically relevant in terms of efficiency. And then if you actually go deep into it in sequence, and this was done together with Ryan Collins at the Talkowski lab at Harvard Medical School, you see that it predict most of this damage that you're seeing is a predicted deletion, even though there's also some inversion. So sometimes the CRISPR is cut and then just inverts. Now in principle, most of it is the total on-target that you would expect. And that, as I told you, goes almost up to 50% with the best guide we have. So this is very good, but we can only go forward if we actually keeps the function of the cells. So to do that, we tested these cells in mice that are immunodeficient. That means they don't have their own immune system. They have no T cells, no B cells, no NK cells. They're like the mouse version of a bubble balloon. And so we use those mice to put in our cells and ask if this normal cell versus edited cells could still generate all blood cell types. And so this first model that we use, NSG, non-skip gamma chain mouse, it has a very slow T cell development. So what it could show at this point is that these cells are able to make both lymphoid and myeloid lineages. So lymphoid C19, T cell marker, myeloid lineage 11B, is a dendritic cell marker. And so in addition, you could also think, well, maybe you somehow selected for the cells that were not edited. Because remember, at most only 50% of them get the genome editing I want. But we did check, if we take these cells from mice, you can see that the genome editing is still there. So you see that the deletion band is still present. So these genome edited cells can robustly contribute to both myeloid and lymphoid lineages of the blood. But then next one, a better model to show that actually T cells can be generated from these edited HSCs. So you turn to this model that's called a humanized mouse. And the way you make a humanized mouse is you take again your immunocompromised mouse, but this time you take a piece of fetal human liver and fetal human thymus. And then you also take, from that liver, you put some in it, and then you also take a piece to sort out HSCs from the fetus. Because in the fetus, HSCs are not in the bone marrow, they're actually in the liver. And so you make this mouse that has human blood, and you have to let it responsibly for several weeks, and now you have a mouse that has human blood. And this model is called the DLT mouse, not for the sandwich name, but for bone marrow, liver, and thymus. And so what happens in this mouse if you put these cells in? Well, we see that they nicely are able to give rise specifically to CD4 T cells. And that's this graph over here. So if you look at human CD45, so a human blood marker to distinguish from mouse cells, you can see that you can give rise to CD4 T cells. And this is across the all mice we did. Okay, so this part of the work will actually be published already, and we were able to honor Boston in our graphical abstract. We're able to honor New England on the cover of that journal. That's the foliage right there. And then, this got some press. This is what Time Magazine thinks we did, a cure for HIV. But this is really what's ahead of us, is to prove functionally that these HSCs that can make T cells, but are these mice, first of all, now protected against HIV. So those experiments are now ongoing. If you have this humanized mouse receive an HIV challenge, are the mice that get the CCR5 deficient HSCs protected against developing HIV? So that's what's ongoing right now. And then this is a part that we're working on right now. And then if you're in a schematic form, if you now want to translate this into people, HIV patients that will edit these cells, and then you can put them back in, and hopefully this will eradicate HIV. Because they now have cells that are immune to HIV, so they'll not get infected themselves, and then they can kill other cells that are already infected. But from a short term, more interesting for us, is that we're now able to, for the first time, systematically interrogate immune cells at a genomics level. So this is just a very quick schematic just to show that if you have your immune cells, T cells could be B cells, could be natural killer cells. If you can now use this CRISPR-Cas9 and you have to modify it to have a specific cell type, you can now for the first time really change genes in immune cells and not just surrogates like other acids. And then that could lead to discover several things. In our case, it's very important because our samples don't get too many immune cells from our maternal-fetal interface samples. So you want a system that works really, really well, and you can interrogate gene function at really this genomic level. And so from in a more futuristic perspective, you can also think that you can now engineer cells for therapy, and they can actually become the third pillar of therapeutics. The first one being small molecule drugs, the second one being antibodies. So now we can actually, for the first time, inject cells into people and use that as a therapy. This is just a case that got pressed in the UK of a baby that got cured of cancer with T cells from another person, where they, not surprisingly, edit the genes in those cells related to the immune system. So this could be the future of this kind of genome editing in immune cells. And so with this, I'll go into the most important section, the acknowledgement section. So I'm going to begin at the beginning, and that means that I would first of all like to thank the 3 professors back in my college, University of Coimbra, that believed in me when I was 18, 19 years old. They let me work in their labs, and they wrote me letters of recommendation, and without them, I would not be here today. I'm sure they would enjoy being here, that's why this is being filmed. But so, it really was in July 2011 that I first came here, and I'm part of the MCP department. You know, we've had our differences, but looking back, I couldn't think of a better grad program to be part of because my classmates are really smart, very supportive of the environment. So, I've been really being spoon-fed during my 5 years of graduate school. And so when I first was applying to Harvard, I had this, romantic image in my mind of this is the best place in the world to do stem cell research in regenerative medicine. And So, we have Doug Galton, David Scanlon, created this Harvard Stem Cell Institute. It was already used in Time magazine. And so I was very happy that I got to be part of this. I was across the ocean seeing these things on the internet. I'm actually here. But so the life-changing moment really in grad school was when I met Jack. And so I remember going to Jack's office and talking to him about my interest in understanding how the body can tell self from non-self, and how we could change that for therapy, change that awareness, if you will, of self versus non-self. And then that became a rotation in his lab, and then I joined his lab. Jack is a charmer. So, and then, many times Jack lured me into free dinners at Courier House, and when we get there, Jack is like, Leo, I printed your lab meeting slides, and I only have like 100 slides for my lab meetings. Let's go over that. So Jack is unstoppable. He thinks about science 24/7. Every time you give a journal club, Jack will drop 10 papers on your desk, and you better read them all and discuss them. So also Jack is not afraid of embracing the latest technologies to explore problems he might see. Here's Jack with his new iPhone 6s. He did not let us eat dinner before he could Instagram, his side of the table. And Jack is always, he knows how to explore, to be surrounded by the best people. And what I really want to say is that, again, Jack comes into lab every day, he's so enthusiastic, he's a kid at heart, and then he really leads by example. So thank you, Jack, I'll really miss you. And in addition, I have another PI, I'm that lucky, Chad Cowan. So Chad Cowan has been amazing. He met with me every single week during my rotation time, and very frequently afterwards. And Chad has a very wide expertise in experimental, so he helps me with the nitty-gritty details when I'm struggling with experiments, as well as the big picture. And then importantly, he's also very, very supportive of all my successes. He established varied collaborations, the various projects we saw, many of them were established by Chad. So it's been amazing help. Thank you, Chad, for having me for 5 years. I don't think Chad knew how much work I was when he first took me in. So also, actually, I have a third mentor, a more direct mentor, Thorsten, which I'm sure provided much of that material at the introduction. So I really have to thank Thorsten for helping me so much in these years. Thorsten taught me how to do experiments, how to learn, how to teach. I actually taught a class together, Scrub 165. There's some students right there, with Chad Cowan is a professor. And also I saw the lab was, again, a great mentor and a great friend. He helped me explore the Harvard campus, as well as abroad. This is in New Orleans. And so, we don't always agree. But I think we got much better at Thorsten's best or favorite word, communication. I think we improved greatly in communication during these past years. So all this has been possible thanks to a terrific environment, so the labs we've been part of. So the Strominger lab is just from a different world. They're just really, really helpful. I love them so much. They share reagents. We have really care about This is in Jack's farm. This is in Jack's office. Sometimes it's really like from a different world. And I think we know we can blame that for, right? Can blame that people who pick the people. And the same thing goes for the Conlin lab. The Conlin lab, again, extremely helpful lab. How many times have I bought Currys and Macs for reagents, for advice, for everything? And all the people I worked with, all the collaborations. It might be a fierce-looking lab at first, but it's actually all very nice people, and they're always there to help you. So thank you guys, Strominger and Conlin lab. And then, of course, I also have to thank my dissertation advisory committee, Rich, John, and Hide, for keeping me in check every 6 months. And then, to be honest, I don't think, or to be realistic, I don't think I'll ever be able to give back to all these people. It's just been so much. So one way to ease my pain, I guess, would be to try to make other people feel that way about myself. So being involved in teaching. And so I was very lucky that I was able to teach both in Harvard College a large course. LS1A is a large biochemistry course for freshmen. 12 out of 13 of these students are in science concentrations. That was their first science class. As well as Scrub 165, a small lab course which Chad as a professor. Taught it last year, it was a great success. This year even more. There's some people right there. And then I got the chance to teach not only at Harvard, but also abroad. So as I already mentioned in the introduction, there's this project called Clubes de Ciencia, which was actually started in Mexico by Ben. He's a grad student in the chemistry department. And then it's being spreading out. So now it's directed in Bolivia by Mo over there, and in Colombia by Carolina, and they're my classmates. At Harvard, the world kind of feels small sometimes. So many good people together. And so it was a really great experience. So I got to thank to have gotten the invitation to the talks and the lab for the students. But really, I think my favorite part was what happened in the lab, or it wouldn't be me, right? So I was able to get students under my direct supervision and I got 3 of them. Brianna Johnson, which you've seen already, Hannah, and Andrew. And no one wants to see live pictures anymore. So okay, so Brianna Johnson did her senior thesis with me, and she's now going to medical school starting this fall. Hannah came here for the summer. She was incredibly hardworking, so hardworking, and in 3 months of work with me, she's actually on one of my papers. And she's coming back this summer to Harvard in June. And for Andrew is my latest student. He's a rising senior. He just got a Herschel Smith Fellowship to also being here during June. He's incredibly smart. He joined my supervision at a time where I'm obviously really, really busy, so he just learns everything at first. It's been amazing. And you know, I'm leaving the lab, but these two will still be around, so even though I'm leaving the lab, my way of doing experiments in a way. So I think that's great. And you know, grad school is also time to find, you know, all sorts of passions, and so one of them was dancing, as again you've already seen, but I was very, very lucky to be part of the Harvard Ballroom dance team, and these are my 3 partners. Carly is actually in the audience today, and then there's Ma and Emily, and so this was a really amazing time. I used parts of my brain that I never used before, and had a great time. You know, all these skills actually came into being very, very useful. I really felt like sometimes I was dancing with the stars, literally. And, you know, finally doesn't go without saying that I also got some special people. I guess these 3 people. So this is Christian, also in the audience. He was my first roommate ever. I was in my parents' house before coming here, and it's from Miami, and they met doing interviews, and he accepted to room with me. And, you know, it's just been amazing, and like, taking care of everything. And then also Patrick. We've been together in pretty much something like 15 different countries. Actually, we just came together from Mexico last weekend. And you know, it's been amazing. And then in addition, also Mo right there, which not only gave me the chance to, you know, be part of Publicis Bolivia, also every time we're on a table, any of these 3 people, we just start talking about science right away. And everyone gets very upset. That's just who we are. And then in addition, also in the first 2 years, Mo and I really struggled together through classes. Sorry, I struggled. He did okay. Right? Great. I was the one. So, you know, thank you very much. Without 3 of you guys, I really would not be here today. And so, you know, I didn't put a title on that slide, not because I forgot, because I don't know if we should be called classmates, roommates, friends, the brothers I never had. I wasn't sure, but, you know, I don't know where we'll be 10, 20, 30 years from now. But I hope we'll all be really famous scientists, so we can keep challenging ourselves, and I can get to show that photo in, like, you know, big conference talks. So with that, I have my official, if you will, acknowledgement slide. So my 2 PIs, Jack Strominger and Chad Conlin, as well as the labs. There's a short list of people involved in the pregnancy immunology project and in the CRISPR in immune cells project. My funding by the MCB department and the Portuguese Foundation for Science and Technology. And then other people are on this slide. Doesn't mean I forgot about you. There is, for example, Joyce over there to help with the facts. Renato, another thing, is here, help with making figures. Bill McCallum, who helped me so much, like sorting out all kinds of things, and Jennifer Shea and Nancy Wheeling. So many, many people. So really, I want to end on a note to say that it's a really magic place. Thank you so much for having had me. And you know, wherever, I don't know where I'll end up, but this place will always feel like home. Thank you for listening, and I'll be happy to take questions. Yeah, Eva.
Great talk. So you showed that data and CPP regulation HLA expression are still not specific for both of us. So what do you think are other factors in the experiments?
Yeah, very, very, very good question. So one thing that we know is that we found a transcription factor that is really specific for trophoblasts called GCM1. So at least in the mouse, this transcription factor is only expressed at the invading tips of trophoblasts. And GCM1 has been reported to regulate CBP alpha expressions, one of the CBP factors. So one idea is that these factors reach upstream of these factors will regulate that. Or maybe it binds to the enhancer directly or to a different enhancer. So currently looking into this. One difficulty I've had, I must say, is that, you know, I show you the gain of function experiments, the loss of function, I also did that. The problem is that if you knock out GATA2 or CBP alpha or CBP beta, the cell just won't grow. And I think it's because these transcription factors have so many functions of these cells that the loss of function has been harder to carry out. So I guess it's two aspects.
So how do orangutans solve the pregnancy problem? Do they have a non-polymorphic HLA-G equivalent?
Yeah, that's a good question. So it's the answer will be funny because I read that if you look at the habits of chimpanzees and gorillas versus orangutans, it seems that orangutans are involved in more long-lasting relationships, while chimpanzees and gorillas are more promiscuous. So the mothers are subject to more allogenic fetuses. So one idea is that somehow when orangutans diverge and this enhancer L could have come, or HLA-G could have come as a response to that increase in allogenicity. And then actually, if you look at HLA-G polymorphism, the highest number of alleles is found in individuals from Africa. And that's because they're exposed to many, many infections. And so this balance of keeping immune tolerance and at the same time fighting infections is a delicate balance. So at some point, HLA-G has been rendered to be non-polymorphic, so it is involved in immune tolerance. But in certain places, such as in Africa, you see more alleles arising. So again, suggests there's been some sort of transition between HLA-G being a classical MHC and becoming non-polymorphic and more involved in immune tolerance.
We got lost also in the beta trend knockout strategy. Why is that important? Why would you want to do that?
Well, so beta-2M is the easiest and safest way to ablate MHC class 1 surface expression. So these cells will become invisible to T cells from the recipient.
With the exception of the case.
Yes. So that's why ideally you could ablate MHC class 1 surface expression and then put in an HLA-G with a linked beta-2 microglobulin, such that you have specific HLA-G on the surface taking care of the NK cell aspect of it, and then you get rid of the MHC class 1 to take care of the T cell aspect of it. From a functional perspective. From experimental perspective, beta-2M is a very nice gene because it's expressed everywhere. Several people have used these guides to test their cell lines for amenability, if you will, of genome editing. Because it's always expressed, you have just a simple FACS experiment that tells you right away what efficiency editing you have. Yes, what's it?
So, great talk. I was wondering along the evolutionary lines. So, with the CNS1 promoter element in FOXP3, right, that's a really beautiful story because if you look for that evolutionary rationale, it basically arises in placental mammals, right? So it's missing from your platypus, but then everything else downstream has it. So that makes a lot of sense, right? And in your story, I was wondering that, where does HLA-G first arise? And then why does enhancer L only really arise in that primate lineage? Or did you just not show us maybe other species that are intermingled and don't have as much of it? Does it have something to do with migratory patterns, the genetic diversity that might require that extra kind of safety net?
No, very, 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 very good question. Very good question. So the CNS enhancement story is very beautiful, and I was almost going to show you as something that we did not do. And that's because HLA-G gene does not have a clear homologue in mouse. And so you can't, unlike that paper, you cannot just go by sequence conservation and see, okay, the highest conservation in non-coding sequence must mean there's an enhancer there. And then you have a very nice kind of parallel between evolution and the function. So in this case, we cannot do that. So that's why we have to take a functional approach. So actually going to functional test enhancer elements across the locus and then delete them with CRISPR. And this could be, a new way of discovering regulation of genes that are human specific. But then in parts of the conservation, I think it's just not clear. So there's a lot of differences between, say, mouse and human pregnancy. So mice have many, many pups, and it only lasts 21 days. So some people argue that there's no such thing as an HLA-G system in mice. It's just that difference. That could tell you why it's in apes but not in rodents, for example. And then between monkeys, you also see differences. So in that case, I think it's just been harder to explain. Because as you might imagine, it's very hard to get placental tissue from chimpanzees and gorillas and orangutans and other tissues that can really look at tissue-specific gene expression. So you have their genome sequence, but not really their tissue expression level. So I guess the answer is I don't really know yet what the true reason is. Yes.
Have you expressed the cell type specific transcription factors that bind enhancer L in a different cell type that doesn't express HLA-G? Would that be sufficient to drive HLA-G expression?
We know it isn't because, for example, 293 T cells that I showed you do not express HLA-G, do not have chromatin looping. They express these factors as well. So we know.
Weren't there cell-specific, like that GATA2 or something that you were
Yeah, so
saying?
the GATA3 is very specific. Basically, also expressed in the blood.
But if you express that in 293 cells, would that be sufficient?
No, it's not enough. Yeah.
It's not.
So the thing is that, we're missing transcription factors, definitely. And so one way we went, so it's always hard to go from DNA element to trans-acting element. So DNA element, you have, it's much more specific effects, so you can knock it out, and it only changes the gene expression. Once you start trying to test for transcription factors, other aspects that are not necessarily involved start arising. Like, for example, if you knock out CBP alpha, CBP beta, the cells just don't grow. And so I see that they do lose HLA expression, but I'm not confident to affirm that that means that I proved that CBP alpha binds to enhancer, which in turn controls HLA expression, because it might be a secondary pathway because the cells don't grow that is affecting HLA expression as well as other genes. So I guess we're still in the search for cell type specific transcription factors that govern this second layer. So one idea would be, if I'm allowed to continue, is that you can do what's called a reverse chip. So normal chip, you have your protein factor, you bind to that, and then you ask where it binds. Reverse chip would be you have the region of interest, then you ask what binds to it. And so CRISPR-Cas9, in theory, you do have a protein that binds wherever you want in DNA. So if you get that to work, technically speaking, there could be a way to discover, in an unbiased way, transcription factors that are binding to these enhancer encoders. So this currently work in progress. Yeah.
Theo, I'm a neuroscientist, so it might be some question, but do you think that HLA-G acquiring this immune suppressing function could somehow have enabled longer pregnancies in great apes? And slightly related question, could you speculate as to whether how other animals such as elephants that have long pregnancies pregnancy paradox?
Yeah, that's a very good question, especially the elephant one, because when I was in India last year, I actually thought about how the elephants, they're pregnant for like over a year, so it's a really long time. So it'd be very interesting. Again, I guess once you go from well-established model systems to other animals, it becomes harder to test or hypothesis. So again, you have nice data sets for expression across all tissues, etc. So it'd be hard to test. But I guess if I can speculate, so it seems from here that it's not so much involved in length of pregnancy, but more in the degree of foreignness. So if you have, say, in this case of the apes, if you have a community where everyone is somehow more genetically identical than in other communities, it seems that, so in orangutans, so like isolated species within the mountains, so they became so similar that an HLA-G system is not as important as for chimpanzees and gorillas that just like going around, and then the mothers will, in consequence, be exposed to more foreign fetuses. And so that somehow arose, but it's the same gene, it's just that it's non-polymorphic, so somehow the evolution that matters. But these are all speculations, but unfortunately, it's even hard to come up with an experiment to test my speculation. It's just hard to get the materials to start with. John?
Simple one. So you kept calling it herb, but do you know what subclass it was? Herb H?
Yes.
Okay, so
So
herb
it's a
H.
human specific.
Yeah, well, there's several human specific herbs. So the herb H was shown by the Shogi lab that Gifford and all review cited. And for enhancers, all herb H elements have K4 methyl or active in pluripotent stuff. And there's recent reprogramming or implantation paper showing that they're required for implantation. Have you looked carefully in human yeast cells to see if HLA-G is expressed or what might be different about the HLA-G promoter that is that enhancer, which makes it almost 100% active in pluripotent cells, how it doesn't activate HLA-G, or is there some sort of turnover of mRNA?
Yeah, no, good question. So we have looked at expression of HLA-G in stem cells, and as far as we can tell, it's not expressed, especially by FACS. There's some papers showing even other cell types HLA-G mRNA expression, but it does not become protein expression. So there might be some post-transcriptional regulation going on here. Because, for example, even in cancer samples, many cancer samples express HLA-G at the mRNA level, but if you do an immunostaining or FACS, it's not there anymore. So I think there is some layer of regulation there. And then, third question, I think, well, so what the stem cells are, it doesn't work that, it seems like. I haven't done the experiments. I don't know if it loops in stem cells, maybe it doesn't loop, so meaning that they might be missing a factor, or maybe the chromatin marker is not just conducive to expression. So I haven't looked yet.
One more question. After that, there's the usual libation in 268. And if you're thirsty or hungry, please come. One more question. Where did I silence it?